Protein synthesis by the greatest group ever. Starring Allie's head. Yeah. Chicka chicka. The first phase of transcription is initiation. In this phase, RNA polymerase binds to the DNA double helix, as illustrated in these gorgeous pictures. That's the RNA polymerase. The polymerase starts to unwind the double helix. The second phase is elongation. The polymerase has now fully expanded the double helix . In the interior of DNA, there are 4 nucleotide bases: cytosine, adenine, guanine, and thymine, being drawn as I speak. As the RNA polymerase travels down the DNA, the polymerase forms a complement of the template strand of DNA. It uses uracil instead of thymine. This complement is a strand of mRNA. A termination sequence in the DNA signals RNA polymerase to stop adding new nucleotides. The RNA molecule is then released, as shown here. Yeah. The first stage of translation is also called initiation. The mRNA molecule, now in the cytoplasm, bonds to an rRNA molecule. This rRNA molecule is made up of a bottom piece and a top piece, which acts like an enzyme. A tRNA is also involved in this step, as it comes and binds to the rRNA at the mRNA site. It's right there. It's here. Elongation is also the second phase of translation. The tRNA molecule has an anticodon of 3 bases, which binds to its 3 complement bases on the mRNA. There's a specific tRNA molecule for every possibility of codon. Each one of these tRNA has a different amino acid attached to it. That is the amino acid, there's the tRNA, and there are the anticodons and codons. While the tRNA is still bonded to its codon, another tRNA comes to its codon next in the sequence. Their amino acids are bonded together by a peptide bond. As each new tRNA molecule comes, the previous amino acids that were on top of the first tRNA are passed to the second tRNA. They are formed into a polypeptide chain. The first tRNA leaves and a new one comes. The polypeptide chain is retained on the active tRNA, but will be passed to the next one and the next one, and so on. The final step of translation is called termination as well. A stop codon in the sequence is next in the chain, and this attracts a release factor, shown here in blue. That is the polypeptide chain. This is slow. The stop codon and the release factor move together, and this causes the polypeptide chain to be released and the last tRNA to be released as well. Then the RNA debinds and is released, as well as the rRNA, the release factor, and the mRNA. This is the end of the greatest project the Earth has ever seen. And this is the greatest ending to any project the Earth has ever seen or ever will see.